or sponge is that? Because I've seen it in the data logger. If we go by one, can we zoom in on it just so I can see what it is?
Garcinia. Yeah, I'm not sure which one that is.
Yeah, I'm not sure. That's what one of Steve's notes.
Mm.
Coral.
Look it up though.
And if you're familiar with the tropical soft corals that are, they're not the fan-shaped ones, but the big kind of elaborate columns with lots of little frills. Actually, about to go above the lasers right now, if you zoom in, that's a Garcinia, just to the right of the heteropolypus.
Mm. Oh, back the...
Yeah, back to the right. We've been seeing them in this depth band, so I'm sure we'll get another one.
I think I saw a moldy cheese sponge back there as well.
Thank you for backing up so that we can get a close-up on
Sure,
the...
which one was it?
So it's just above the twig, just directly to the right of that mushroom coral, that pink blob.
Blob. Looks like a... From afar, it looks like a little Chrysogorgia puff, but that's not what it is.
No, it's actually got pretty solid texture to it. It's a soft alcyonasian coral. So it has sclerites, but they're not joined together to form this thicker skeleton, and it doesn't have like a central axis made out of the gorgonid, just this kind of softer tissue. This one's actually pretty condensed down in this current. They can stand pretty tall when they inflate with water fully, and then they have these elaborate kind of frilly polyps around the edges. Yeah. This one is probably Garcinia julie-peckardi, but there are a couple out here, and it's a group where Garcinia is another one of these groups from the Atlantic. That a lot of corals out here that have this soft coral morphology have been called Garcinia, and some of them are not actually in that genus. So we're doing some work to try and tease out who is who.
Thank you for the zoom in.
Yeah, sorry for the rough video there, the sideways of the current.
It's a very strong current.
Yeah, I need to turn my head into it next time. It'll be better.
The stick in front of it is Swiftia simplex, which is a very widespread, very common species of Swiftia. It's one we've done some genetic connectivity work in, and at least in the coastal U.S. waters, it looks like there is just one single population coast-wide.
Bring your head to the right a little if you want.
Do you want me to pause here or slow down? 0.2? You're a bit...
I think we're all right.
Okay. Okay, as long as you're okay with that.
Yeah.
Just keep an eye on targets.
We've got all this fractured rock here. And one of the reasons they peeked up over the top is we've been looking for bedrock and have yet to find any. So Guy was commenting earlier that it may just be completely broken up in this scarp area, but that's one of the reasons we're going to keep occasionally peeking over the top, is just to see if we can find actual bedrock.
Do we want to?
Other than the
yeah, let's
do that now.
That.
Okay.
Let's go. Yeah, let's do that now before.
I'll bump Argus up slope. We're going to go 050, or we'll just go at an angle. So 015.
Right.
And
bridge nav.
What are the candelabra-like looking corals? Not the Paragorgia, but the white ones that have sort of like candlestick.
Those are Isidella tentaculum, and they almost always have this candelabra form. Although some of these single bamboos may actually be Isabella that just have not branched yet. The nice thing about Isabella when they're in these candelabras is it makes them a very distinctive species to ID. And they are species Tentaculum. And if you look at the base of some of them, you'll see these long frilly polyps that are just clustered around the base. And they're a coral that can get.
Go ahead.
They can get quite large. I've seen specimens that are more than a meter across, and they can be quite old at that size, more than 200 years old.
So as we're moving up, if we see one of these Isadellas, maybe we can zoom in on the base, like the one that we're coming up on. Is that okay?
Sure.
Dan and Steve, and we can look at the base and see those long polyps.
With respect to the lasers now, the one to the left or the one above?
The one to the left, whichever is easiest. I think you know which one we're talking about.
Zoom there, Steve. No, that's not the guy, is it?
It probably is. So some of these are looking a little beat up. And so it may be that something has eaten those polyps. Like if you see the base, that has completely bare skeleton.
And there's a skeleton right to the skeleton to the right.
Zoom in a bit more there, Steve. That's good.
And a lot of these corals can also pump water in and out of their tissues. And so sometimes they just have them retracted and not kind of flowing in the breeze.
All right, zoom out.
We'll see if we can get one that has its frilly tentacles out.
Thank you, Steve.
My pleasure.
What is the orange looking one above the Isadella that we just passed? I mean, orange yellow.
That's another heterocony sponge. So they can actually come in two colors, the white and then also this yellow color.
Zoom in there a bit, Steve. Good, thanks.
Oh, that's pretty.
Yeah. They end up with the same, overall structure and everything as the white ones, but yeah, they're just yellow. We're not really sure why that happens.
See all the brittle stars?
And there's a little crab back there as well.
Yeah. And then one of those petal sea cucumbers perched right there, too. There's so much to see when you just stop and zoom in.
Yep, little tableaus everywhere.
Yes. Thank you for zooming in.
What's that?
Targets have been fairly stationary. They're sorting out a heading change.
Roger.
There is that Isabella right there, just to the left of the lasers, has the long tentacles at the base.
Oh, yeah.
Oh, cool.
Zoom in on him,
Let's
Steve.
look at it.
Mm.
Yes, it's interesting that some of these corals will have their polyps out and some do not.
Mm-hmm. So with these big bamboos, they can't really retract their polyps all the way down into what we call a calyx, as much as, say, the Swiftia or the Paragorgia can. They can kind of pull them tighter. And here you can see they're all being blown in the current to one side. And just on the right-hand side of the screen, we've got another octocoral. This is the white kind of frilly mound on the lower right-hand corner. That's
Yes,
Peristonella. That's a primoid.
Huh.
And the kind of pink color you're seeing on it is from the brittle stars that are all over the surface.
I think we're starting to make that move, so might have to pick up.
Roger.
Yeah, in this one
Yes,
shot, we've had 3 different families of octocoral.
and I could really see the long, frilly polyps on the bamboo coral.
Yeah, that's what gives them the name tentaculum.
It's good to have a coral biologist specialist on watch with me.
Yeah, I need someone who knows all the other invertebrates.
Sorry, I'm not that person.
I'm sure you will be by the end of the expedition.
Well, maybe a little bit. I feel like I have learned quite a lot.
So I would like to sit next to, either Erica or Tom and just like absorb the fish and invertebrate IDs.
Yes. We just went past is that another 8 case?
Looks like it,
Yep.
Yeah.
Meredith and Lisa, can I ask if either of you is hearing a clicking noise over the SPL?
I'm not hearing a
Sometimes.
clicking noise regularly. I'm hearing a little occasionally that sounds like a microphone maybe, but not dramatically.
Sounds like a keyboard.
Yeah, it kind of sounds like a keyboard. I don't know if there's something, if somebody has something they're fiddling with, or if maybe your microphone's hitting a button on your shirt, or so.
Oh, it might be my microphone hitting a zipper on my sweatshirt.
That could be it. Is your microphone kind of hanging down onto the sweatshirt?
Yeah.
Yeah, maybe if there's a way you could maybe move the zipper, or if you put something softer underneath it. That
I
might
just
help us.
tucked it in.
Beautiful, thank you. Yeah, I
That's
think that was
probably
it.
why I'm not hearing it.
Yeah, that's why I asked, so we could narrow it down.
Current's really ripping. Oh,
Yeah, it is.
you're like full lateral just to stay straight.
Yeah. Is
About,
it hard?
yeah,
Is
75%
it
of the
hard?
power to hold it.
You can see the tether too, just
Pegged over.
Pulled so far out.
The tether, our tether's doing the same thing as the corals. It's strumming in the breeze.
All right, I'm starting to see that this is kind of flattening out, which matches our bathy, and we're looking at it in sonar. So we'll continue as long as you want, and then we can get back down to that steeper slope.
Yeah, so if we just follow along the contour line, and then we can pop on down again.
Okay, sure. So we'll follow along the contour that we're currently at towards the northwest.
Yeah, maybe follow that for how much further to 3C along this line?
It's roughly, let's see, 600 meters away.
Oh, okay. Yeah, so let's just follow along here maybe for 50 meters and then go back down
Sure thing.
again. Thank you.
Bridge now. Cancel this move and we'll do 50 meters bearing 325. Thank you.
It's interesting that as soon as it flattened out, the animal assemblage has slightly changed. Like suddenly the corals are not as abundant. Still have sponges. All it took was just coming over that lip.
Short and squat stuff up here.
It's a big crab.
Big crab.
Yeah.
Is that a pom-pom anemone in the foreground?
Yeah,
Yep.
I think yeah, I think so. They've been seeing them. So Meredith, what do you think about, since being on the top here and we're in a completely different sort of assemblage, would it be worth it to run a transect up here as a comparison?
Yeah, it wouldn't be bad. How far have we come since the last transect?
Yeah, let me take a look. We're 150 meters from the end of the last one.
Yeah, we could run one up here and then drop back down.
Okay, so we'll just continue on that 50-meter step. I'll just keep going until we get to 200.
Okay.
Following along this contour.
That sounds great.
So we'll keep our. It's up to you, Dan, if you're.
Yeah, I'm good with that.
Yeah. So we'll be going 325. Taylor Ann, let's call this.
Start
Here.
All right. It's transect 3.
Thank you.
Thank you.
And just look downstream and see how steep it is here. We can maybe do it.
Yeah, it's not so.
Oh, we're supposed to.
Not so steep.
325, you said.
5, yeah. And you want this to be 200 meters transect?
Yes,
200 meters.
Okay, great.
We're sort of doing the same old, same old.
Big crab.
Oh, yeah. Who wants none of this transect?
Cooking.
So these quantitative transects, for anyone who's following along with us right now, are a way we use to compare habitat up and down the coast in these deep sea coral and sponge habitats. The pilots are flying at a relatively constant elevation as much as they can. We're going to cover about 200 meters. And this video will be reviewed at the NOAA's Southwest Fisheries Science Center, where all of the fish and invertebrates that are bigger than those 10-centimeter lasers in the center of the screen, they'll be counted and identified. And because we use the same method up and down the coast, we are able to compare between the different habitats, which gives us a better picture of the types of habitats and environments that the corals and sponges prefer to live in. And we use this data for a variety of different things, just characterizing the habitat itself. We provide it to fisheries managers, and it also feeds into habitat models to help better predict where we might find corals and sponges.
Might have to bump the speed a little too, or I'll slow down when you can bump it up. Looks pretty benign.
That was a great explanation. And if those of you who are listening, you decided to take a transect here, because if you were listening earlier or watching earlier, we were along more of a wall, and we saw a lot more coral species. And now that we're at the top of this ridge line, we're seeing more sponges. And so we can compare, as Meredith was saying, across the different physical habitats to see what helps determine the type of assemblages, and that's just groups of species that you will find on the deep sea floor habitat.
I'm just gonna pause the transect here while the boat catches up.
Okay.
We're just gonna pause the transect here for a second while the Argus catches up to us.
We're moving with the current now.
Yeah.
Racing along.
Yeah, we're sailing downstream. We were holding to 0.4 there. Just we didn't had the ship at 0.3, so I got out of the box there. Okay, here we go again.
What is that? What is this sailing term when you have the wind in your back?
Oh, that I don't know.
Yeah.
I'm not
It's,
a sailor.
So it's not upwind, but we have the, yeah, I forgot. We have
Downwind?
Just now, no downwind, but I think it's, yeah, sorry. I'm obviously
I know
not
what
a
you mean.
sailor either.
Yeah, the computer's applying the brakes here to limit Herc speed to 1/2 knot.
Oh yeah, I see that. Keeps throwing on the.
Doesn't want it to be a racing ROV.
Still walking speed, but if you walk looking straight at your toes, it looks like you're going really fast. Look through a straw at your toes and you're really going fast. That's basically what we're doing.
And the occasional heteropolypus up here and the occasional small bamboo coral, but definitely a thinner assemblage up here on top than down over the edge.
Crabs are just massive.
Yeah.
Windward.
Windward.
Yes. No.
You're breaking up again, Lisa.
The deep sea soul just
I-
going off screen on the right.
Okay, I was thinking of running or beam reach.
What was that? You said running or?
Beam, B-E-M,
Oh, right, beam.
beam reach. Do I still sound horrible?
It's still breaking up pretty intermittently.
Yeah, what
Oh.
kind of headset do you have a headset or earphones? What kind of microphone setup are you using?
The same one that I've been using all of these days. It's and this is the first time it sounds so crackly. I'll get out again and just come back and see what happens.
Yeah, but the way it's
There's a big furry head.
I'll see.
Yeah.
I can see.
That'll be a short spine at that size.
Hello?
Hey, Lisa.
Hello.
Oh.
Hey. Third time's the charm.
Yeah. Do you want to try move? Maybe I don't know how far away the microphone is from your mouth, but maybe try moving it a little bit closer and see if that helps us.
Okay, now it's really close.
Okay. Well, you're definitely louder, which is good. You were a little soft before.
Okay. Nobody ever says that I'm soft. I'm usually too loud.
Sorry, I know it's frustrating.
Yeah.
Oh no, this is just in many ways it's great that we can still continue to do field work this way.
Yeah.
And I think we all would prefer to be sitting together in the control van, but I think we're really, making do and doing a good job. So thank you for making this happen.
Yeah, thank you for your patience.
So
Gotten a
there's
little more
Isabella.
bamboo coral in the last couple minutes, but it's still around.
Yeah. Proving us wrong. So are those little squat
So tonight's dive is
anemones?
on a fault scarp feature here at Santa Lucia Bank, which we're hoping to continue to find some steep terrain in.
What is a fault scarp?
Although right now we're doing a transect up on the top of this feature, just to get a little contrast of the habitat with the steeper slope down just to the south of us.
So a fault scarp is where you have a fault line where there are two microplates on the floor or the ground that have moved in opposite directions, and where one side of the fault has moved vertically up and the other one down. And sometimes there's also horizontal
The
movement.
big crab, not more sponges. Right at the beginning of this transect, I thought I saw a Ladincunia sponge. I've been keeping my eyes out for those. They're what we call a demo sponge, which is just a different type of sponge, one that tends to be less brittle and tough than the glass sponges. And this one looks like a little moon with big craters on it. The one I saw was green, although we do see them in two colors, green and brown.
Meredith, are you hearing us on SPL?
You might have to slow him down a bit.
Bridge now.
Oh, well, all right.
I
We
can
got
look
some
down,
tilt
yeah.
down.
Meredith?
Add one more step, 20 meters. Thank you.
Yeah, maybe Meredith can't hear us.
Yeah, you might want
Also,
to, if someone's on science chat, could you
I'll send
message her? Thanks.
I will.
I just figured out I can't hear you, and I'm about to restart my.
Okay, good.
Hey, we can talk about her now.
You were doing great narration, but Dan asked, "What's a false scarp?" And you just talked about something completely different.
That was our first clue.
Yeah.
I thought she was just ignoring me.
Yeah, and I thought she was ignoring my explanation too, and I thought maybe people can't hear me.
Yeah.
No, I just was like, why is it suddenly so quiet? No one else is talking, and because everyone could hear me, and I couldn't hear anyone.
So did that make sense what a fault scarp is?
It did, thank you.
Yeah. So what we were hoping to see along this fault scarp is because of the vertical movement that which created the wall. We were hoping to see along that wall or that steep slope that would have formed great habitat, or at least at the top where we would find some bedrock. But finding bedrock on Santa Lucia has been very hard thus far.
Yeah, I haven't seen any bedrock up here.
Yeah, we didn't see any on our former dive. It was as if we were in a continuous talus field. This is sort of fractured rock that you typically find at the bottom of a mountain. And we climbed that mountain and we got to the top, and it's hard to figure out where all of that fractured rock had come from because there was no bedrock.
Kind of looks volcanic to me.
Yeah. But this is supposedly not a volcanic bank in origin.
Oh, yeah, the one from the other night. This one's different.
Yes. Yeah. Yeah. You mean from Davidson? Davidson is definitely volcanic.
Not the Davidson one. What was the one before that? The Palm. The Palm one that looks volcanic.
Yes, the palm. Yeah. That was more on the escarpment, the Santa Lucia escarpment.
So we're nearing the end of the transect.
Okay.
Just pause at the end, and then I don't know if you want to head back down slope here and try to get back down to the steeper slopes.
Yes. Yes,
Sure.
we'll do that at the end of the transect.
Right there.
Get back to where all the corals are.
Yeah.
Yes. And if we see anything as we're going down, we can stop and zoom in. And if, we were going to collect some fractured, we got a request from Dan Geiger of the Santa Barbara Natural History Museum to get a coarser sediment scoop.
Coarse sediment scoop, okay.
So we can use the scoop.
You want to do it up here?
Yes.
Okay.
And if we find it in an area with shell hash, so a lot of pieces of what were formerly clams or any other invert that forms a shell, I think that's what he would really like to have.
Sounds good. Okay, Taylor Ann, we can mark the end of transect here.
Alrighty. End of transect.
And the ship is slowing to a stop, so we'll kind of settle out and find a good scoop spot.
All right.
quick shrimp popping around.
Yep.
Okay, so Dan is saying, try and find an area with salt and pepper looking sediment
Salt and
for the
pepper.
scoop. That seems to be more coarse rather than mud.
You can tell us if you see anything on as we're scanning here.
I'm looking.
Yep.
The only salt and pepper I see is sort of rock.
It looks pretty muddy between the rocks right here.
Yeah. Well, we can just move forward a little bit.
Yeah, we got a bit of leash to play with here as we look around.
Okay,
Just
whatever works for you all.
and then if you don't have anything there, we can step around. But
It's just I'm not somebody who sits in the bus facing backwards.
Sorry, I turned 180 to
No, I'm just
put my head into
I'm
the
teasing.
breeze.
I'm teasing. So it's just I was thinking like what does this remind me of?
It's, usually I say the ROVs do not back up because our thruster trail of dust always catches up to us, but in this case we're driving with our reverse camera.
Daniel is saying there are some areas with whitish spots near the rocks.
Okay, so stop
Right here.
and right where the lasers are to the right. Maybe that's what he means.
Keep an eye and see if he chimes in again.
Yeah.
See right by the baby
So-
fish.
Yes.
Zoom in there if you want, Steve.
fortunately,
Sure.
So I'm just going to ask Daniel because he's on. Is this okay? Right where the baby fish is. We will try to not scoop him up.
I think
He says-
the small thorny head might need to look that out of the way.
Yes, he says yes. So we can do a scoop here. Ooh, and we'll mark it with the, because I imagine we have to back up, get the scoop, but we can mark it with the little sea star here, the bat star.
sure.
And looking open that. I have no idea where the scoop is. It could be in the starboard box. It could be in the front box.
I think it might be that knot as because we'd have 5 core.
Oh,
there it
yeah,
is.
that's it. Okay.
I was like, is that a 6?
Get out, right. Get out. You can zoom in on the little baby rockfish there while we're getting the scoop out.
So small thorny head, when they first land, they're totally gray, and then they get redder and redder. And the last thing that turns red is their little pectoral fins.
There's a little
Some
gastropod.
samples in that box already.
What's an E, Taylor Ann? Is that a rock with a sponge or just a sponge?
It's a rock and a sponge.
Okay.
Yeah.
Hopefully cemented down.
Oh, there he goes.
And it's
Oh.
a baby long spine thorny head.
Yeah, the third spine. Whoop. So Dan, would you want us to scoop up that larger long snail with the scoop? Is that what you're asking?
He's just juggling it.
What's that?
Talk about the scooping up the snail. Do you want the snail?
I think Daniel is communicating via side chat. He says, "Can you get the larger long snail?" And that's the only snail I can see, unless somebody else sees another one.
Sorry, I was confused
Yeah,
because
there's
there's
That's
two
the only one
Dans
I'm
now.
seeing as
one in
well.
the chat and one in the van.
Yeah.
Can you zoom out, Steve? Thanks.
Yep, he said yes. Scoop up the larger snail along with more sediment.
Come on. Sorry, I've got to regrip here. Spastic. All right, where were we? Are we here by the...
A little star.
one more time. So I don't like the grip.
I'm trying to handle things with oven mitts while wearing a blindfold.
Yeah. Those long coral jaws there. Mr. Waters' favorite, and we don't really care for them a lot. They're great for cutting coral, but trying to pick your nose with a baseball bat. Okay, you can zoom in a bit, please. I heard something about, oh, I see the little snail you want? The snail
Yep.
and the star? Is that right? Oh,
I think the snail knows we're coming.
it's definitely not mud.
Like, yeah, it's little rocks there.
You don't have to get the sea star. That was just to mark the spot.
All right. I'm pushing the vehicle around now. How full are
Clearly
we
a very
meant to get this thing?
thin.
That's good.
So, and then we're meant to filter it out, are we? Or just put the whole thing in the box?
Yes, just put the whole thing in the box.
Copy. Okay, you can zoom in there, Steve, and the minute's going away. Using another camera to. Yep. I'll put it back.
Oh, just the upper left. There's a little fish, and I think it's a brotula.
There's a crab coming for
Yep,
us.
that's a ruby nose brotula.
Where
The
are
little
you
fish
seeing
that
it?
just underneath the little rock, the rocklet, you'll see just a little fin sticking
Oh,
out,
yeah.
and you can just make out an
Yeah.
eye. They hide like that. They're very hard to see. You'll frequently just see a tail sticking out from under the rock.
Good eye.
Okay.
Coming in. What was the sample number on that?
Sample number 45.
Got it. Thank you.
And that's in starboard F, right?
Correct.
And what kind of brotulid is that, you said?
I think it's a ruby nose brotula. I'm going to make a note for Tom to zoom back through the video and look tomorrow morning. That's one of the fish he's always on lookout for.
Oh,
The little
that is
guy
so
hiding under the
cool.
rock to the left.
Yep.
Mm-hmm.
Yeah.
Huh.
Can we zoom in any more, or this is as far as it
Yeah,
goes?
if you give me a second here, I'll center him up and we can zoom some more.
Thank you. So you got the time for and everything, Meredith, to send to him?
Yep, I'm looking at it
Great.
right now.
Well, I guess if
That's
they're
full
always
zoom.
under rocks, thank you. If they're under rocks like this all the time, it must be very hard to get a good view
Yeah.
of them.
Oh, wait, I could
Beautiful.
be wrong. It might actually just be our friend, the short spine thorny head, or long spine thorny head tucked in under. Now that I've got a good zoom on it. Centered up.
And based on the large pectoral fin there, and the
Yeah,
that
it's sticking
pectoral fin's
out.
black. Yeah.
Yep. I was so hopeful, but it was the brotula.
Yes.
But I'm going to send him the time anyway, just so he can check.
Yes. Have the expert make a
Yep.
finding, a determination. But thanks for the zoom in.
No problem.
Ooh, the brittle star is moving in.
Mm-hmm.
It almost looks like Gumby, the way they was moving.
Yep.
Okay, so where were we? We were going to go downslope again, correct?
Mm-hmm. Yep, we were
Okay.
going to drop back down to that 850 meter
Yes. Contour.
Sure.
contour.
Can send us down there. So we'll go 150 meters at 245. It'll be mostly downslope, cutting it a little bit.
Okay, thanks,
Bridge
Rennie.
nav.
So do I sound any better now?
You sound a lot better.
Step 150
Yep.
meters, bearing 245.
Great.
I've been fiddling with some settings, and I might have done something that changed it, or maybe something out of my control. But it's sounding good right now, so I'm not going to touch it.
I'm going to say you did it.
I'll take the credit.
Yeah, you always should when whenever it's put out there. Does the Herc really have a reverse camera that you use?
Yep. There's a aft cam, there's a port cam, starboard cam.
It's not HD, but if you look at satellite feed 3, there's the camera.
Okay.
We call it Herc butt.
It's not elegant,
That's
but
a technical term.
it's technical.
Is that a soul there?
Yep, that's the deep sea sole.
Nice pattern, color pattern.
They're one of my favorites. I always really enjoy seeing them because they're so distinctive. And I always find it really fun how if you're working in a very vertical canyon, you'll see them just stuck pancaked to the wall. With the question of how exactly do you stick there?
Yeah. Maybe they have suction.
It's got to be something like that. Or maybe they can use their scales for friction or something, but you'll see them stuck to just nearly vertical walls.
Might be a question for Jess. Our 4 to 8 pilot, she's into the
Adhesion
Bio,
properties of different
Yeah.
materials.
Oh, that's
biomechanical
right.
adhesion. Don't know the proper term.
The Brisingid in the lower left.
There's a yellow morph of a heterocony, right?
Mm-hmm.
Do we know if the sea star wasting disease affected any of these deep sea stars, or was it mostly the ones that are shallower and in the warmer water?
To my knowledge, it was the ones that were shallower and in the warmer water because it seemed to be
Yeah.
quite temperature linked.
Yeah.
Unfortunately, actually, I was out diving a couple weeks ago and saw signs that it might be back. There were definitely some sea stars that had it, which is worrying because we were actually starting to see some of the big sun stars again. But time will tell. Now, happily, we're going into our cold.
Excuse me, what did you just say? We're going halfway into our
Oh, we're going into since we're going into winter when the
mm,
water gets really cold, that should help mitigate any effect of the disease around, hopefully.
was that a dive for work, or was it for pleasure that you dive?
That was for pleasure. In my spare time, I'm a dive master for a local dive shop here on Bainbridge Island. So I was out diving with them.
That is so
We've got-
cool.
Got a local beach that's really good shore entry, so we dive there quite a bit.
So you dive shallow where you get wet and you dive deep where you stay dry.
Yep. See, I get asked frequently, do I dive to look at my corals? And I say, nope, unfortunately. Everything I work on is way deeper than you can get to on scuba, with like one species exception. And that's the, we get one of the species that all sea pens are octocorals, and so there's one of the sea pens that has quite a broad depth distribution, and it's the Tylosarcus gurnii, the orange sea pen, does get grouped with the deep sea corals, but you can find it all the way up to 3 feet of water. And then there's one spot in British Columbia, or a couple spots in British Columbia, where some of the Paragorgia come up shallow enough that you can see them on scuba. And then in Alaska, some of the Primnoa do the same. That's the red tree coral. Now we're getting back into a more coral zone.
Mm-hmm.
Just at the top of it where it gets steeper.
Yeah.
Well,
Yeah, so we can follow along. So I think we're on, are we at 840 meters?
Mm-hmm.
yeah,
And
we
so
are.
that's a little bit lower than the contour we were on previously, correct?
I can go double check. I think it's a bit higher.
But we can,
Let's see.
oh yeah, it's high. Yeah, it's higher. Yeah, so we can follow along here. What do you say to that?
Just watching along at home, we've been talking a lot about deep
Zoom in there for a
sea
second,
corals
Steve.
and sponges, and we're looking at a nice assemblage of these deep sea bamboo corals here. Corals and sponges are, of course,
That's good.
animals, although ones that are quite what we call basal to the tree of life. They're very old, very simple compared to vertebrate. And normally when we think about corals and sponges, people tend to think of big tropical coral reefs, but corals are actually found worldwide in the deep ocean and in cold water habitats as well. And we're interested in studying them because they form part of what we call essential fish habitats. So these are these islands of biodiversity in the deep sea that are valuable to fish as habitat. And that includes fish that we're interested in eating once we get up into shallower depths. Down here at 850. Oh, can't hear you again.
Yes, can you hear us, Meredith?
I don't think it's updating.
So taking pictures?
Yeah. I don't think Meredith can hear us. So
I think you're right.
yeah.
Try log in again, maybe.
Yeah. So I'll send her via side chat.
I'm back. I can hear you now.
Yes.
I
Okay.
need to get on with it. Huh?
Yeah, no, it's just the USBL is also being
Right.
funny. So
So,
Yeah, so
Ship still moving, or
yeah, we're still moving.
No.
So
stop him up for a minute?
yeah. Bridge nav.
Meredith, do you,